Hello everyone, welcome to our channel Biological Concepts. Today's topic of discussion is miRNA versus siRNA. Here we will learn the differences and similarities between miRNA and siRNA. For CSR NET, ICMR NET, GATE and various other exams, this topic is very important. Now before discussing miRNA versus siRNA, we have to know what is miRNA and siRNA and why this is so important topic. Now, miRNA and siRNA are non-coding RNA that play important role in gene regulation. Nowadays, these serve as novel therapeutic agent for the treatment of a wide variety of diseases, including cancers and many other infectious diseases. Now, let's discuss the differences between miRNA and siRNA. So, let's start. First is full form. The full form of miRNA is microribonucleic acid, whereas the full form of siRNA is short interfering ribonucleic acid. Second is the mechanism of gene regulation. miRNA regulates the gene by various ways. First is by translational repression. Second one is the degradation of mRNA. And the third one is by endonucleolytic cleavage. This third way, that is the endonucleolytic cleavage, is very rare in case of miRNA. It is occur when there is a high level of complementarity between the miRNA and mRNA. Whereas in case of siRNA, the mechanism of gene regulation is only by endonucleolytic cleavage of mRNA. Third one is the number of strands. In miRNA, it is single-stranded, whereas the siRNA is double-stranded. Now, how you will remember? We know that double-stranded is shortly written as DS. So, siRNA S and for double-stranded D. So, this siRNA is double-stranded, that is DS. Okay. Next is structure. These miRNA are 19 to 25 nucleotide RNA with 2 nucleotides 3' overhang. The structure of miRNA, that is a microRNA, includes double-stranded stem, a terminal loop and 2 unstructured single-stranded segments. Whereas the siRNA is around 20 to 25 nucleotides long RNA with 2 nucleotides 3' overhang. So, in case of both miRNA and siRNA, this 2 nucleotide 3' overhang, is common. Okay. Next is the nature of RNA. The miRNA is endogenous, whereas the siRNA is exogenous. Now, why this siRNA is exogenous? The siRNA is an exogenous double-stranded RNA that can either be chemically synthesized and then directly transfected into the cell, or it can be generated inside the cell by introducing vectors that express short hairpin RNA, which are the precursor siRNA. Okay, that's why these siRNA are exogenous, and in this way these are produced outside the. Next is the occurrence. The miRNA is seen in all plants and animals, whereas the siRNA can be seen in lower animals and plants, not observed in mammals. But in miRNA, this miRNA is observed in the mammals. So, miRNA is observed in the mammals, but siRNA are not observed in mammals. Next is the prior to dicer processing. Prior to dicer processing, miRNA is in its precursor RNA form, that is the pre-miRNA form, containing 70 to 100 nucleotides with scattered mismatches. The pre-miRNA exists in a hairpin loop structure. So, this miRNA prior to dicer processing is in its precursor mRNA or pre-miRNA form. Okay. Whereas prior to dicer processing, siRNA is double-stranded molecule containing 30 to 100 nucleotides. One important thing is that in case of miRNA, another protein, that is dorsa, is also required. Dorsa is also required for processing the miRNA. Whereas in case of siRNA, this dorsa is not required. So in case of miRNA, both dicer and dorsa is required for miRNA processing. Next is the complementariness. miRNA is partially complementary to the mRNA, typically targeting the 3' untranslated region of the mRNA. UTR means untranslated region. So this is the unique feature of miRNA, that is, it targets the 3' UTR region of mRNA. Whereas the siRNA is fully complementary to the mRNA. Next is pairing with the target. How miRNA and siRNA pair with the target mRNA. miRNA pairs with many mRNA targets imperfectly and inhibit the translation, whereas the siRNA pairs with only one mRNA target. Okay, so miRNA pairs with many target. M means miRNA, that is the first is M, then is I. So miRNA many target, M for many. Okay, and this siRNA pair with the mRNA perfectly, that is, it is highly specific. Next is requirement of gene silencing. AGO protein only, not necessarily the AGO2. Any protein from the organoid family can participate in the miRNA mediated gene silencing. Whereas in case of siRNA, only the AGO2 protein, that is the organoid protein 2, is required. Not any other kind of AGO protein is required for the gene silencing. So in case of miRNA, any kind of AGO protein can participate in gene silencing, whereas in siRNA, the AGO2 protein, this very specific, is required for gene silencing. Next is the clinical application. miRNA has various kinds of clinical application. First is therapeutic agent. Second one is drug target. And the third one is diagnostic and biomarker tools. So miRNA serves many kinds of clinical application. First is therapeutic. It acts as a therapeutic agent. It can serve as a drug target, or it can serve in diagnostic and biomarker tool. Whereas the siRNA serves only as therapeutic agent. So in short, miRNA is microRNA, siRNA is small ribonucleic acid. The miRNA has multiple mRNA target, whereas siRNA has one target. And miRNA has multiple type of clinical application, whereas siRNA has one kind of clinical application. Okay, these are the differences between miRNA and siRNA. Now let's discuss the similarities between miRNA and siRNA. First is, both miRNA and siRNA are non-coding RNA. Both are post-transcriptional regulator of gene expression. Both are incorporated into the RNA-induced silencing complex, that is RISC. When the siRNA incorporated into the RISC, it is termed as siRISC, whereas when the miRNA is incorporated into the RNA-induced silencing complex, then it is termed as miRISC. Okay. Now both are closely related in their biogenesis, assembly into the RNA protein complexes, and the ability to regulate gene transcript. Number 5 is, both are generated by dicer, a multi-domain enzyme of the RNase 3 family. Both miRNA and siRNA need to be unwound before they can be assembled into the RNA-induced silencing complex. These are the similarities between miRNA. If you like this video, please like, share and subscribe to this channel. Thank you.